How the deletions of autism-associated genes SHN-1 and NMR-2 affect the rate of synaptic transmission in C. elegans, an Etch-a-sketch stop-motion animation. Kind of. First, let me give you some background on autism spectrum disorders, or ASD, and how they relate to synaptic function. Autism spectrum disorder is characterized by social and learning deficits, repetitive behaviors and patterns, and other changes in behavior. These changes can be a result of many symptoms of ASD, such as changes in rate of synaptic transmission. Additionally, every change that contributes to ASD is caused by different possible combinations of genetic mutations. Since there is such a wide variety of genetic mutations associated with autism and such a wide variety of symptoms resulting from those mutations, there's still a lot we don't understand about the disorder. In previous literature, changes in rates of synaptic transmission have been observed, but it is unclear what mutations contribute to what changes. In my research, I studied Caenorhabditis elegans, or C. elegans, which are microscopic worms that are good models for genetic disorders such as ASD. To observe how certain gene mutations cause changes in synaptic transmission, I used one strain of C. elegans with the gene SHN-1 knocked out and one with the gene NMR-2 knocked out. SHN-1 is a homologue of the gene SHANK3, which is a scaffolding protein that contributes to synaptic formation. Mutations in SHANK3 are commonly seen in people with autism. The other gene I studied, NMR-2, is an ortholog of GRIN2B, which is a protein-coding gene for a receptor. Mutations in GRIN2B are also commonly seen in people with autism. To look at changes in rates of synaptic transmission in these worms, I exposed them to a drug called aldacarb. Aldacarb is an acetylcholinesterase inhibitor, which causes paralysis in worms faster if they have a faster rate of synaptic transmission. When acetylcholine, the square molecules depicted here, is released into the synaptic cleft, it binds to receptors at the postsynaptic neuron, and one of the things that this causes is muscle contraction. Soon thereafter, acetylcholinesterase breaks down the acetylcholine and the muscle contraction ceases. However, if that part of the process is inhibited, such as aldacarb does, then acetylcholine stays bound to the receptors, causing more and more muscle contraction until paralysis eventually occurs. With a faster rate of synaptic transmission, the buildup of acetylcholine reaches the point of paralysis sooner, which is what I recorded in my data collection. To run the assay, I added aldacarb to the plates of the 5 different strains, 1 positive control, 1 negative control, 1 normal strain, and the 2 mutated strains. Then I transferred a group of worms in the L4 stage, or the stage right before adulthood, to the plate containing aldacarb. After that, I timed how long it took for each worm in each plate to become paralyzed, since a faster rate of paralysis is indicative of a faster rate of synaptic transmission. This assay was repeated many times until I had tested around 40 or 50 worms from each strain. Here's the survival curve of paralysis calculated for the N2, or unchanged wild-type worms, that I could compare to the mutant worms. As you can see, the positive and negative control show that the assay was effective, for the positive control worms that were sensitive to aldacarb died at a much faster rate than the N2 worms, and the negative control worms that were resistant to aldacarb never became paralyzed within the 500 minutes I tested them for. As for the worms with the SHN-1 knockout, they became paralyzed at a slower rate than the N2 worms. Meanwhile, the NMR-2 knockout worms became paralyzed at a faster rate than normal. These data showed that, in C. elegans, a deletion mutation of SHN-1 related to the autism candidate gene SHANK3 causes a slower rate of synaptic transmission, and a deletion mutation in NMR2, which is related to the autism candidate gene GRIN2B, causes a faster rate of synaptic transmission. More research is required to understand how these changes are manifested in humans, but this project does suggest that synaptic transmission rates could be playing a vital role in autism, and that mutations in different autism-associated genes cause different types of changes, leaving us with even more questions than we had from the start. But that's what research is all about. Special thanks to Dr. Monahan and my research and biology class for helping me throughout this process. Thanks for watching.